Part 1. Please find a quiet place to sit or lie and to listen to this audio. Once seated or lying, close your eyes. Focus your attention on your breath. When you're ready, inhale slowly. Let the air fill your lungs. And then, exhale slowly. Let the air escape from your lungs. Inhale again. And then exhale again. Notice how your stomach rises as you inhale and falls as you exhale. Rises as you inhale and falls as you exhale. If your mind wanders, don't be concerned. Just gradually shift your attention back to your breath. While you continue to inhale, focus your attention on the air rushing into your nostrils. Imagine the tiny hairs in your nostrils bending in the air, like flimsy trees during a violent storm. Now orient your attention to the scents that waft in the air. Perhaps you can smell a powerful aroma, like a perfume or flower. Or maybe you can smell only a faint scent, so faint that you're not sure this scent is not merely a figment of your imagination. Part 2. So how do you recognise and differentiate the scents and aromas that float in the air? How are these scents or aromas converted to information that your brain can understand? And how does this set of circuits, called the olfactory system, differentiate the vast array of scents that you can smell, such as fresh lavender, burnt toast, and wet dogs? Part 3. The scents or odours in the air are actually molecules. As you inhale, you can imagine these molecules enter your nostrils, like mice scampering into a gap in the wall. Perhaps you can visualise these molecules as small blends of Lego pieces or short coils of ribbon. These molecules then attach to special receptors at the back of your nostrils. To clarify, imagine you shoved a powerful microscope up the nose of a friend. You could then examine the lining of these nostrils, sometimes called the olfactory epithelium. You might notice this lining comprises two main categories of cells. The first class of cells merely support or strengthen the lining. Without these cells, the lining would be fragile and vulnerable. This epithelium would collapse. The second class of cells, in contrast, are the olfactory receptor cells. To some extent, these olfactory receptor cells resemble a broom with long bristles. The bristles are called olfactory cilia, a synonym of hair, but are technically the dendrites of neurons. These bristles, or cilia, protrude into the nostril. The horizontal platform to which the bristles or cilia are attached is the cell body and is embedded within the lining of the nostril. And finally, the long handle is the axon that extends inside the head towards the brain. Don't be concerned if your image is not entirely accurate. Any image of a tiny broom will suffice. As you maintain this image of a cell that resembles a broom, with the bristles inside the nostril and the handle extending to the brain, repeat the term olfactory receptor cells. Olfactory receptor cells. Olfactory receptor cells. Don't worry if you can't remember this term later. Simply repeat this term now as you imagine the broom. Part 4. How do these olfactory receptor cells detect the scents or odours in the environment? Well, on the bristles or cilia that protrude into the nostril are tiny receptors, like tiny combination locks. But then when you zoom your focus on one bristle or cilia and examine these tiny receptors more closely, you realise these receptors actually resemble a skipping rope that weaves into and out of the cell 7 times, The skipping rope is actually a protein composed of a sequence of amino acids like tryptophan, glutamine, proline, lysine, alanine, glycine and tyrosine. Now shift your attention to other bristles or cilia poking into the nostril. On each bristle, you will see more of these tiny receptors, each resembling a skipping rope. And each skipping rope weaves into and out of the cell 7 times. But these skipping ropes, these tiny receptors, are not identical to each other. Instead, the precise shape varies across these receptors. And the precise order of those amino acids also differs across the receptors. Indeed, as you rapidly scan the various receptors across your nostrils, you notice about 300 distinct clusters of skipping ropes. They're 300 distinct shapes, if you like. If possible, try to imagine many different shapes of these skipping ropes, even though each skipping rope weaves into and out of the cell 7 times. But why do the nostrils comprise so many distinct skipping ropes or receptors? How do these receptors differ from each other? Well, each receptor tends to attract a distinct class of chemicals. For example, imagine you can watch a chemical, such as a chemical with a chain of 5 to 3 carbon atoms, float into the nostril, like ash in the wind. You might notice this chemical binds to receptors, or skipping ropes with a specific shape, but not to receptors with another shape. Then imagine you can see another chemical, such as a chemical with 7 to 9 carbon atoms, also float into the nostril. Now this chemical might bind to receptors of a different shape. Finally, imagine inhaling the fumes of a dish you cooked, such as a beef stew or a vegetable soup. Feel the fumes penetrate your nostrils. After you inhale the steam that emanates from this dish, many classes of molecules float into your nose, each of which bind to a distinct class of receptors. Consequently, most scents actually activate more than one class of receptors. Part 5. So what happens after a molecule binds to a receptor? How does this event translate to the conscious awareness of a scent or aroma? In essence, after a molecule binds to a receptor, a protein inside the cell, but attached to the membrane, is released and floats into the cell. And this protein is technically called a G protein. Perhaps you could imagine a molecule that resembles the letter G, disconnect from the membrane and drift into the cell. And as this protein floats into the cell, channels or canals in the membrane open, culminating in action potentials or sparks of energy. Now the details are not important now. Only focus on the notion that molecules, after binding to the cell, evoke an action potential like a spark of energy. In other words, when bound to molecules, these olfactory receptor cells, the tiny brooms embedded in the lining of nostrils, ignite sparks of energy. But to where do these sparks of energy travel? That is, to where do the axons, that is the handle of the broom, tend to end? Well, this spark of energy travels down the axon and eventually reaches a region of the brain called the olfactory bulb. Actually, the olfactory bulb is nearby, sort of just behind your eyebrows, at the bottom of the cerebral cortex, the top half of the brain. The olfactory bulb then classifies the scents. For example, very sweet aromas might activate one set of cells, whereas just moderately sweet aromas might activate a nearby but distinct set of cells. Whereas the scent of flowers might activate a different set of cells altogether in the olfactory bulb. Finally, the olfactory bulb then transmits action potentials to many other regions of the brain, such as the uncus, the amygdala, the hippocampus, and the orbitofrontal cortex. You don't need to memorize these regions now. You merely need to appreciate that each region provides a distinct role. For example, The amygdala determines whether the odour is pleasant or unpleasant. The hippocampus evokes past memories in which individuals experience the same odour. So many regions of the brain connect the scent or aroma, a scent or aroma that is categorised by the olfactory bulb, to other evaluations, interpretations and memories of the past.